Assalamu alaikum. In this video we discuss the functional anatomy of the kidney, the tubular system. The tubular system or nephron consisting of renal corpuscle and the renal tubule. Renal corpuscle or renal glomerulus discussed in the previous lecture, while now we'll discuss the renal tubule. Renal tubule consisting of single epithelial cell. These single epithelial cells have two membrane, luminal or apical membrane that is the lumen, and the basolateral membrane that is the interstitial fluid. The luminal membrane consisting of numerous microvilli that decrease the surface area, while the basolateral membrane consisting of numerous mitochondria. Number of the mitochondria differ in the different part of the tubular system, and it is very important in the active transport for processing of the glomerular filtrate for reabsorption and secretion of the filtrate between the luminal and interstitial fluid. Between these cells there is lateral interstitial space or paracellular space, and there is a tight junction between these single layer epithelial cells. Now we discuss the different part of the tubular system. These, after the filtrate pass through the Bowman capsule, we start the proximal convoluted tubule, loop of Hilli, distal convoluted tubule, and the collecting duct. The glomerulus is discussed in the previous video. Now we discuss the tubular system. So we start with the proximal convoluted tubule. It is also begin in the cortical part of the kidney, and also have citrate part that descend in the medullary part. This part of the collecting system have numerous mitochondria for the active transport, and then the solute or filtrate descend to reach to the loop of Hilli. That loop of Hilli start in the medullary part of the kidney. The loop of Hilli contain two parts: thin descending limb and the ascending limb, and also the mitochondria. It's different in between two parts of this loop of Hilli. This is the descending limb and this is the ascending limb. And the ascending limb is also have two parts. This is the thin descending limb of loop of Hilli, and this is the ascending limb. The ascending limb have two parts: thin descending and thin ascending and thick ascending limb. The last part of thick ascending limb of loop of Hilli ascend in the cortical area and reach to the glomerulus of the same nephron and pass in between the afferent and efferent arteriole in a specialized area. This specialized area called juxta glomerular apparatus, which contain specialized cell called macula densa. We'll discuss later on. There are different, as we say, there are different type of nephron. This is cortical nephron or superficial nephron, and the other type is the juxtamedullary nephron. And difference is the long loop of Hilli that dip deeply in the medulla. After the loop of Hilli, there is distal convoluted tubule, and the distal convoluted tubule also contain numerous mitochondria. After that, the solute parts and the filtrate pass to the collecting system. Numerous nephron collecting in the same collecting nephron duct that descend from the cortical area to the medullary area, and also larger and larger collecting system coagulate and unit together to forming in the lower part of the medullary part of the kidney to forming the collecting inner medullary collecting duct, and they form the duct of Bellini or papillary duct that descend into the minor calyces of the kidney, then to the pelvis.